Hello, my name is Matthew Graham,
I'm Josh Klinnert.
and today we're going to be talking about the pentose phosphate pathway. So let's get started. Here's some general information. It has a major role in NADPH production, and it's an important shuttle molecule. NADPH plays an important role with the reduction of many things such as fatty acid oxidation, nucleotide biosynthesis, cholesterol biosynthesis, neurotransmitter biosynthesis, and it's also very important in detoxification of the cells. You may also know the pentose phosphate pathway by some of their more common names, which would be pentose shuttle or the phosphopento or phosphogluconate oxidative pathway or the PPP. There's some very interesting health facts. It also helps prevent oxidative stress. The location of the pentose phosphate pathway is in the systole, and there's been small amounts found in the endoplasmic reticulum. The overall reaction is glucose 6-phosphate plus 2 NADP plus molecules and a water molecule to yield ribose 5-phosphate, 2 NADPH, and 2 CO2s and 2 H+. A fun fact is that there is actually a 7-carbon compound that is produced, and it's rarely seen in the human body, so it's kind of interesting. Also, there's tissue distribution. The pentose phosphate pathway is mainly located in lipid synthesizing tissue such as the liver tissue, adipose tissue, adrenal glands, and mammary glands. Let's talk about some of the products and their fates. So with the NADPH that is produced, it can go to reducing double bonds within other molecules as well as carbonyl groups. The ribose 5-phosphate can also be used as energy metabolism in shuttle molecules such as NADH, FAD, and coenzyme A, and information molecules such as DNA and RNA. This is actually one of the 5 fates of glucose 6-phosphate. It's important to cellular and metabolic capabilities because the pentose phosphate pathway supplies the NADPH for most everything in the cell, all the way from fatty acid biosynthesis to cellular detoxification. It helps reduce the overall oxidative stress. That's the main route for glucose metabolism, and it has a major role in xenobiotics, which is a glucuronic acid drug detoxification system. This allows for scrambling of glucose carbons, and it also provides nucleotides via ribose 5-phosphate for production of DNA and RNA during cell division. The 3 phases of the pentose phosphate pathway. The first one is the oxidative phase. The tissue distribution, some tissues actually only carry out the oxidative phase. It is the conversion of glucose 6-phosphate to ribose 5-phosphate. The reactions includes glucose 6-phosphate dehydrogenase, lactonase, and 6-phosphogluconate dehydrogenase. The second phase is the non-oxidative phase. Reactions include ribulose 5-phosphate isomerase and ribulose 5-phosphate isomerase. The third phase is the scrambling phase. This allows for metabolism of excess ribose 5-phosphate. The reactions include transketolase 1, transaldolase, and transketolase 2. An increase in the scrambling phase actually decreases the chance for cancer because all of the ribose 5-phosphate is being used to synthesize RNA and DNA during dividing cells. Here's a comparison of the pentose phosphate pathway right next to glycolysis, kind of comparing the shuttles, the energy products. With the pentose phosphate pathway, the shuttles are NADPH and glycolysis is NADH. There's a big difference between those two. The pentose phosphate pathway actually yields no ATP, and glycolysis does. The products for the pentose phosphate pathway are ribose 5-phosphate, and it has multiple exit points, and with glycolysis there's only one product. The reactions in specific, the oxidative phase is glucose 6-phosphate dehydrogenase. It actually plays an important role in regulating the pathway. You take G6P plus NADP plus to yield 6-phosphoglucono beta lactone and an NADPH and a H plus. And after that, lactinase spontaneously happens on its own. It takes 6-phosphoglucano beta lactone or delta lactone and a water molecule to yield 6-phosphogluconate and a H plus. The last step is 6-phosphogluconate dehydrogenase. This clips off a carbon as a CO2 molecule. It takes 6-phosphogluconate and NADP plus to yield ribulose 5-phosphate and NADPH and CO2. In the non-oxidative phase, there's 2 steps. The ribulose 5-phosphate isomerase moves a double bond to oxygen to the end of the molecule. It takes ribulose 5-phosphate and transforms it to ribose 5-phosphate. And the second step is ribulose 5-phosphate epimerase. This affects the stereochemistry of the third carbon. It takes ribulose 5-phosphate and changes it around to xylulose 5-phosphate. In the scrambling phase, the first step is transketolase 1. This is actually where the production of the 7-carbon molecule is formed. It takes xylulose 5-phosphate plus a ribose 5-phosphate to get a cetoheptulose 5-phosphate and a G3P. The transalkylase takes that 7-carbon molecule and combines it with G3P to form fructose 6-phosphate and erythrose 4-phosphate. Transketolase 2 takes xylulose 5-phosphate and erythrose 4-phosphate and combines them together to get fructose 6-phosphate and G3B.
Alright, now since we've gone over the overview of the pentose phosphate pathway, I'm going to dive into some of the enzyme specifics. So these first 3 enzymes I'm going to go over are going to be the enzymes that are involved in the oxidative pathway, which again is important for producing molecules that are used for reduction of molecules of other organic molecules. So the first enzyme I'm going to go over is glucose 6-phosphate dehydrogenase. The substrates for this reaction are glucose 6-phosphate and NADP+. So glucose 6-phosphate dehydrogenase is going to take these 2 molecules. It's going to form 3 products. The first of which is 6-phosphoglucano delta lactone. The second is going to be NADPH and hydrogen molecule. The cofactors for this enzyme are going to be NADP+, but NAD+ has also been used to be a cofactor. So the specific type of reaction is going to be a redox reaction. Regulation of this enzyme is going to be NADP+, which allosterically regulates it. Hormones such as insulin have also been shown to regulate this enzyme. They can activate or deactivate the enzyme. Some other interesting details: glucose 6-phosphate dehydrogenase specifically alters the hemiacetyl group on glucose 6-phosphate and transforms it into an acetyl group, which then forms the product of glucose 6-phosphoglucano delta lactone. A disease that is specific to glucose 6-phosphate dehydrogenase is going to be glucose 6-phosphate dehydrogenase deficiency. This is caused by mutations in the glucose 6-phosphate dehydrogenase gene, such as glucose 6-phosphate dehydrogenase A minus and glucose 6-phosphate dehydrogenase Mediterranean alleles. And these mutations cause deficiency in glucose 6-phosphate dehydrogenase, which then leads to an insufficient amount of NADPH to be produced through the conversion of glucose 6-phosphate into 6-phosphoglucano delta lactone. The NADPH that is normally produced through this reaction is used to maintain a reduced form of glutathione, which is then used to prevent oxidative stress within the cell. Many cells have other means of maintaining a reduced form of glutathione. However, red blood cells must rely on the pentose phosphate pathway for this reduction. Without the reduced glutathione, oxidative free radicals will build up in the cell, causing damage to the cell membrane and possibly cell lysis. This type of cell lysis can be referred to as hemolytic anemia. The treatment for this kind of disease is going to be mainly prophylaxis, avoidance of triggers, which are fava beans, infections, and medicines/foods that have high oxidative properties. In the acute form of hemolytic episodes, patients can be given blood transfusions and dialysis to facilitate the removal of dead cells. Another enzyme is going to be lactonase. The substrate is going to be 6-phosphogluconate delta lactone and water. The products from this reaction are going to be 6-phosphogluconate and a hydrogen ion. Cofactors, this enzyme doesn't really have a major cofactor that plays an important role in the reaction. This type of reaction is going to be a hydrolysis reaction. It can also occur spontaneously without the enzyme, but it obviously takes a while, and so it's not really favored without the lactonase. 6-phosphogluconate dehydrogenase. The substrate is going to be 6-phosphogluconate NADP+. The product is going to be ribose 5-phosphate, carbon dioxide, and NADPH. An important cofactor is going to be NAD+, which helps with the reaction, helps the enzyme activate its active site and bind the substrates. The type of reaction is going to be an oxidative decarboxylation. So those were the three enzymes that were important for the oxidative phase. These next two enzymes are going to be important for the non-oxidative phase. The substrate is going to be ribulose 5-phosphate, and the product is going to be ribose 5-phosphate. Again, this enzyme is not going to have a major cofactor. The type of reaction is going to be an isomerase of a ketose to an aldose. This reaction is reversible, so it can go in both ways. The second enzyme is going to be ribulose 5-phosphate epimerase. The substrate is ribulose 5-phosphate. The product is going to be xylose 5-phosphate. This enzyme doesn't have any important cofactors. The type of reaction, this reaction is also going to be a catalyzation in the interconversion of the hydroxyl group on carbon 3, giving it a opposite configuration. Okay, these three enzymes that I'm about to go over are going to be important for the scrambling phase. So the substrate is going to be xylose 5-phosphate and ribose 5-phosphate. The product is going to be cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. The major cofactor important for this is going to be thiamine pyrophosphate. And the type of reaction is going to catalyze the transfer of a 2-carbon group from ribulose 5-phosphate to a 5-carbon group. Transaldolase. The substrate is going to be cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. The product is going to be fructose 6-phosphate and erythrose 4-phosphate. No major cofactors for this enzyme. And this reaction is going to catalyze the transfer of an aldol linkage. Transketolase 2, the substrate is going to be xylose 5-phosphate and erythrose 4-phosphate. The product is going to be fructose 6-phosphate and glyceraldehyde 3-phosphate. This cofactor is going to be the same as transketolase 1. It's going to be thiamine pyrophosphate. So the regulation of the pentose phosphate pathway is going to be that first step, and it's going to be the conversion of glucose 6-phosphate to 6-phosphogluconate delta lactone via the enzyme glucose 6-phosphate dehydrogenase. This is considered the first committed step of the pentose phosphate pathway. Glucose 6-phosphate dehydrogenase is further going to be regulated by the availability of NAD+ compared to the concentration of NADPH. Hormones can also play an important role in regulation. Insulin, for example, has the ability to activate glucose 6-phosphate dehydrogenase and thereby engaging the entire pathway. The modes of the pentose phosphate pathway. The pentose phosphate pathway can be used specifically for 4 different metabolic situations. The first one is going to be cell division. Requirements for this mode is going to be more ribose 5-phosphate than NADPH. This is because dividing cells require more ribose 5-phosphate for synthesis of DNA precursors. The mechanism for this mode is going to be the majority of glucose 6-phosphate is converted to fructose 6-phosphate and glyceraldehyde 3-phosphate. These molecules are then used to form ribose 5-phosphate. 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate are converted to 3 molecules of ribose 5-phosphate by transaldolase and transketolase 1. In this mode, the scrambling phase is highly used compared to the oxidative phase. The second mode is going to be used primarily for the maximization of NADPH production. Requirements: there's an equal emphasis on the importance of both NADPH and ribose 5-phosphate. The mechanism for this mode is that glucose 6-phosphate is used to form 2 molecules of NADPH and 1 molecule of ribose 5-phosphate. Ribose 5-phosphate can then be converted to fructose 6-phosphate and glyceraldehyde 3-phosphate, which can then form glucose 6-phosphate and go back through the oxidative phase, producing even more NADPH. In this mode, the oxidative phase is highly used. The third mode of the pentose phosphate pathway is going to be fatty acid synthesis. Requirements: more NADPH is needed than ribose 5-phosphate. The mechanism: first, the oxidative phase of the pentose phosphate pathway forms 2 molecules of NADPH and 1 molecule of ribose 5-phosphate. Then ribose 5-phosphate is converted into fructose 6-phosphate and glyceraldehyde 3-phosphate by transketolase and transaldolase. Finally, glucose 6-phosphate is resynthesized from fructose 6-phosphate and glyceraldehyde 3-phosphate by the glycogenic pathway. The fourth mode of the pentose phosphate pathway is going to be used primarily for ATP synthesis. So requirements are going to be NADPH and ATP. Mechanism: ribose 5-phosphate formed by the oxidative phase of the pentose phosphate pathway can be converted into pyruvate, fructose 6-phosphate and glyceraldehyde 3-phosphate derived from ribose 5-phosphate into the glycolytic pathway rather than reverting to the glucose 6-phosphate. Pyruvate formed by these reactions can then be oxidized to generate more ATP, or it can be used as a building block in a variety of biosynthetic pathways. And so an important way to trace the pentose phosphate pathway is through labeling glucose at C1 and C6 positions with different types of isotopes. This is important because it can be used to designate specific tissue types such as adipose tissue, mammary glands, and adrenal glands. This can also be used to differentiate between the pentose phosphate pathway and glycolysis. This is because the C1 position is oxidized to carbon dioxide in the pentose phosphate pathway in the TCA cycle, whereas the C6 position is oxidized only in the pentose phosphate pathway, allowing for the differentiation of the two pathways.